biology skills background builder. The first essential concept in this unit will be the scientific method cycle. You can see on the cycle, the picture on the screen, the cycle is outlined. You can start by making an observation. You state a problem, you identify the variables, formulate a hypothesis. From there you can design an experiment which will collect data, analyze the graphs, and draw a conclusion. We'll cover this in depth a little bit later on, but you have to understand that it is a cycle, it is a way of thinking, and that is the first essential concept of this unit.
So in order to understand the scientific method and experimental design, you need to understand this first concept here of what a variable is. Variable can be defined as this. This is something you should have on your definition sheet. A variable is something that can be changed in an experiment. So there's lots of examples, but we've listed a few here for you. So you've got temperature, weather, pH can be changed in an experiment. The length of something can be changed. Time can be changed. There's almost an infinite number of variables that are possible in an experiment.
There are going to be three types of variables that you'll be putting on your vocab sheet here. The first one is going to be independent variable. It is that variable that we intentionally change as we do an experiment. In this experiment here, we're growing plants. That's not the independent variable. The independent variable is the one we're going to change. In this case, temperature is changed. So that would be your independent variable.
The second variable that you need to consider in an experiment is the dependent variable. This is the variable that is measured as a result of what happens or what you do to the independent variable. So looking back at our plants again here, I'll make this a little bit bigger. Now we've circled the part of this experiment that is the dependent variable. You change the temperature as the independent variable, and then you measure the growth in the plants. So there's a lot of different ways you could measure the plant growth in this picture. We could look at the height of the plants, the color, the leaf width, the root length. There's many different dependent variables you could measure, but we can clearly see that there has been a change as a result of the independent variable.
The final variable you'll be putting on your sheet will be the variables that we're going to be called controlled variables. These are the ones that we intentionally keep the same across our experiment. Using our plant experiment that we've shown you so far, here's the one where we grew it in the hot and the cold, but there are other things that we had to keep the same so that we know that our results are going to be valid. We needed to use the same soil, the same amount of water, the same type of water, the same time of watering. We use the same types of pots, the same ventilation, kept in the same location. If we didn't control these variables, they would likely change the results, which was our dependent variable.
Okay, the last two terms which are related to each other, so we have them together, is the relationship between a control group and an experimental group in your experimental design. The control group is a part of your experiment in which you attempt to imitate conditions that mimic normal. So this plant was grown in as close to normal a condition as possible, whereas your experimental group is defined by the independent variable. In this case, we were changing temperature with the hot and the cold. So you need to have a control group against which you can compare the results that happened in your experimental group, or experimental groups, I suppose, in this case. And that's it.